Okay. I see that we're starting to get people in. Hi, everyone. I'm Laura, also known as Lolly on the server. And today I have the pleasure of introducing Dr. Jeff Brender. He is a staff scientist at the NIH and a beloved member of the BioCore community. Today, he is here to talk to us about the Optimus Bind project that explores protein design problems. And it's a wonderful opportunity for folks who are interested in protein folding. Without further ado, I hand it up to Jeff and I will mute myself.
Okay, thank you for having me. I'm glad to be here. And what I'm going to talk about, as you said, is some problems and opportunities in protein design and possibly a project that for people who are looking at projects that we kind of can collaborate with together. So as we know that protein-protein interactions are very essential to cell. They control many things, reactions within it. So they're kind of the control point of many processes. So what happens when something goes wrong? So what happens when you have a mutation that affects protein-protein binding? We'd really like to know what the effect of this is on protein-protein binding and all the processes that go that are downstream from that. We also want to know if we can perturb this protein-protein interaction network towards our own ends, which is part of the philosophy of protein design, which is changing proteins, engineering them to achieve things we want, either to stop them from doing something that we don't want them to do, or to get something to do something that they do either better or to engineer totally new functionality within it. So I'm not going to talk too much about the goal of this because I think it's fairly obvious. One good example of this is with COVID and the binding of SARS CoV-2 to the receptor. So we really want to know what mutations and variants are the ones we have to look out for. So it would be really good to know what mutations are likely to change SARS CoV-2 binding to ACE2. And we really want to know this as quickly as possible. So ideally, as soon as the protein is sequenced and then we find the receptor, we would like to know what mutations we need to screen for in the population. So there are ways of experimentally doing this. One of the most popular ways is by yeast display, and where you tie the genetics and the genetics and the protein structure together. So you have a protein sequence that you want to look at. You introduce mutations in at a few sites. I think it's generally 6 for yeast display. You're limited how many mutations you can incorporate or sites you can incorporate on. You have a air-prone bases at that site. And then you screen by fluorescence-activated cell sorting for the ones that bind to the surface. So in this particular setup, the protein is displayed, hence the term yeast display. It's bound to the outer membrane of the yeast cell. So it is in position to interact with another protein. You watch the protein in. You have proteins that bind to it. The ones that don't bind to it have only one signal, and the ones that do bind strongly have 2 signals. And so then you can use iterative processes of sorting by fluorescence activated cell sorting to separate out the enrich them in the high binders and deplete them in the low binders. Because after every cycle, you can recover the plasmid, sequence it, and you can find out what mutations are associated with the ones that bound. So as I mentioned, you can only incorporate a few sites on this. So if you have, as we saw in the SARS-CoV-2 protein, like the Delta variant, has something like 26 different mutations on it, that some of these interact with each other. So you can't scan for them individually. You have to have pairs of them, and this makes the process more complex. So it would be good if we had a very fast computational mechanism that could scan all the mutations of a protein quickly and test all the interactions, likely interactions between different amino acids so that we can know what mutations and, more importantly, what combinations of mutations are likely to have an effect. So I said the Delta 1 is a good variant of this. All these mutations appeared singly very early on in the COVID epidemic. It's only when they started appearing together that we actually started seeing the effects of the Delta variant. So we need to think about a way of looking at this computationally. And the most obvious method is what I call the brute force approach, or the physics-based approach. This is based on molecular dynamics. Here, you start with initial atomic model, because the interactions are very precise. It should ideally be an experimental model. You calculate the forces on each model. You move them according to these forces, and then you repeat this process over and over. And this gives you both a picture of the energetics and the dynamics of a protein. So for this particular project, this is very good for many things, but it has a problem, and that's very slow. It can take days to do this for a single mutation, whereas we want to do this for thousands and thousands of mutations. So we need something that's much faster. So people have developed a new method called Foldx. Rosetta, which you may be familiar with, works on a similar method where they don't look at the dynamics. They have a fixed backbone and they allow the side chains to move. And they also make another approximation in that instead of allowing the side chains to move continuously, they fix them into only a few discrete conformations. And kind of justification is that most conformations of the side chains of a protein are forbidden. There's kind of basins, and then it hops between basins. So we can kind of discretize it by only looking at the basins and then setting it to a single conformation for these. And this is called a rotamer. And the rotamer library is all the different combinations that can exist in there. So the backbone is fixed, the side chains can move, but only to a few positions. And so what this allows you to do is it allows you to remove those clashes that occur when you try to put a new amino acid in. Now, this has some problems because you can't get rid of them completely, both because you're using discrete conformations and you're not allowing the backbone to move. So what Foldx does, and I kind of call it Implied Dynamics, is it tries to scale by mobility. And it actually, in Foldx, it scales by the solvent exposed area, or how much of it is on the surface of the protein, which is not the same thing, which also does cause some problems. So as you can see here in the structure of protein, there's regions which are relatively free to move down here in the blue, where you can see that there's disorder in the protein. And there's other regions which are kind of locked in. They're conformationally restricted. So we can scale all the interactions kind of by this sigmoidal type functions, because when we're scaling unfavorable interactions, we're also realizing that the favorable interactions are also not likely to be there as strongly. And we can go even further by changing as you see in the upper right here, the function of the terms that we use. So when we have a restricted motion, we use the actual repulsive force, which has a very sharp cutoff when the atoms get too close. Whereas when they're free to move, we have a more relaxed dependence on the distance. So this is fairly fast, but not too fast. It can take several minutes, or depending on size of protein, maybe an hour to do a mutation, which is better than days. But it has a drawback, which is accuracy. So when we look at the Foldx versus experiment, it's really okay. It's not good. It's not great. You have a correlation of about 0.4 to 0.5 with the experimental binding free energy values. So we wanted to look and see if this is work that I did with Yang Zhang in the University of Michigan, if there's another way that we can do this, which is not using this physics-based approach. And this is the program we developed. It's called BindProf, and this is kind of the flow through it. It seems fairly complicated, but I'll walk you through it. The key to it is the idea that similar proteins that serve the same function will be related together. So they'll have the same distance constraints and the same energetic constraints. And so we can use this relationship similar to you do a sequence alignment, but doing it in space. So we start off with a library of interfaces. We have something like 56,000 interfaces. For each one, we then align the protein that we want to look at with one of the complexes from this library. And we then kind of basically, which is like a superposition of the protein structures, the best possible one, but optimized specifically for the interface. And then we score this alignment based on how well this matches. We then apply a cutoff, which is important because if we have the cutoff is too loose, the proteins are not related, and we can't really establish a relationship between the two of them. If it's too strict, then we don't have many points for comparison, and we get bad statistics. From this, then we form a sequence profile similar to what SciBlast would get. So we can see in the first one, this one was too dissimilar. We eliminated it. And the bottom two are kept because they have similar interfaces. From that sequence alignment, we get the frequency of how often the amino acid appears at the position of the mutation, and then we can get a free energy from that simply from the probability that the frequency that shows up in this sequence alignment derived from the structural alignment versus the background frequency of how often that amino acid appears. So this is a very simple equation, although there's a lot of subtleties that get into it that I'll get into later. And so this is a very simple method. Surprisingly, it does very well. So it gives an accuracy basically very similar to Foldx, even though once we have formed the profile, it only takes a few microseconds to calculate the actual potential. So once we calculate once for the protein, we can do all the mutations very quickly in milliseconds, which means that we can scan for thousands of mutations. And as we go through here, it's probably actually better than that because it excels at finding favorable mutations, which are really the ones that we are looking for. So it's similar when we look at all mutations, but it is better than ever than all the other techniques that we tested at finding the favorable mutations, even more better at finding ones that are very strongly binding. And it doesn't do too bad at the unfavorable, which are most of them, but those are the ones we're not that concerned with. So it's a very fast method. It's very conceptually simple, and it kind of is also complementary. It succeeds in the cases where the physics-based methods don't necessarily work. So we have two different methods. We have the physics-based methods and the profile methods. They are good at different things, and they're good at different contexts. So physics-based methods are good at high-precision structures, where we have a locally rigid protein, and that there are a few analogs that we can find in the protein database that we can base this profile scoring off. When we have a low precision structure, like if we're predicting the structure from something like AlphaFold or Rosetta, or that we know that the protein is dynamic, we can't use one structure, and we have to do the scaling, the profile becomes better. And it's also, the requirement is we need many and diverse analogs. So these two are complementary to each other. So the next question is can we combine them? And so when we use machine learning with these two methods, and then we combine them with other scores, which measure how well we predict each one will do. So I mentioned we need many and diverse sequences. So we look at the measures of that, statistical measures of that, such as the entropy of the alignment and the conservation score, and number of sequences we have in it. And then we look at how accurate the structure is based on the RMSD of the alignment and the R value of the crystal structure. We can combine the two of them, and then we have more kind of indirect measures, which are based on the solvent exposure and how it changes and other features that we hope to add more to, and whether, for example, whether it's a small to large mutation, which we expect that things will move apart, and we don't expect the single point structure that is used in Foldx to be very accurate. So we have kind of three basic things. We have the single point Foldx potential, the profile score that we developed, and then we have another set of coarse-grained physics-based potentials, which only work on the residue level, that kind of balance it out and then provide a physics-based method, but one that is optimized for low resolution. And when we do this, we get a pretty high correlation score of 0.83, which I think is the current best at this problem, even though it's, I think, 6 years old now. And we think it was pretty good. What it then allows you to do is we can use it not just to predict the binding of proteins' response to mutations, but we can use it to actually design new ones, which is basically taking the process and iterating it over and over, doing kind of a random search, or not quite random, of amino acids at each site. So here's an example where we have a protein complex, or we knew the structure of it, which is XIP and SMAC, which is an important protein involved in apoptosis and cancer, and we redesigned it so it had only 50% of the identity to the original sequence. And you can see this, the crystal structures compared between the two of them at the interface. I think blue is the design sequence, the crystal structure of it, and red is the original one. So they're basically almost identical. And the binding of it was very close to the original one as well, even though we changed half of the amino acids in the protein. So where this is going with the collaborative project, the current program, which is called BindProf, it doesn't have a server. It's not really open source, and nothing's really available for it. So it's kind of dead to the academic world. New techniques are now possible. There's more data. There's more things that I have thought on. I'll mention two of them that could be improved. There's many more, but I don't want to necessarily get too deep into this talk. And also, we want to expand the number of questions so we can have mutant impact on not only the protein-protein binding, but also the protein stability, which is a similar problem in many respects with a few additional details. And also to look at small molecule binding, not just of proteins. It's really to the question of druggability, whether we can drug this protein or not, or how much we expect, for example, in cancer, how easy it is, or with bacterial resistance, how easy it is to resolve resistance to a drug. Or more generally, predicting the effect on phenotype. So if we have those three together, and then we can do some work with the basically working from the protein-protein interaction graph itself using some elements of graph theory, we can start, and people have done this, there's a kind of prototype in the work of predicting what the phenotype of the mutation change will be. So I wanted to get, so I'll give two examples of two things that could be changed. So I said the program was fast, but there's a long step. You have to go through 56,000 interfaces and align them. So the alignment for each one is pretty fast, like around a second or so, but there's 56,000 of them. So it would be really good if we had a method that could get rid of obviously bad interfaces, which is like 99.9% of them at the start. So we're only scanning for a small number at each time. So I've been thinking of a few methods based on geometric hashing, or actually topological invariance of protein structure, which is kind of, I guess, another way of encoding the 3D structure of a protein, or specifically the interface, into a one-dimensional string that we can match very rapidly and eliminate the ones that we do not think will fit. And that would be kind of a paper in itself. And it would be a very big improvement of the program, because this around 8-hour time to search through all these interfaces kind of frustrates some things. Another thing, which is more I mentioned there's some subtleties when we go through this with changing this multiple sequel alignment to a free energy value. So we're dealing with the two probabilities here, but the probabilities depend on the sampling. So we can think of it as we're sampling along a phylogenetic tree, as you can see here, and oftentimes what will happen is we'll have a big cluster, usually around human sequences, not lamprey here, where they're highly overrepresented. And since we're just doing, if we do a straight count, these will skew the results away from what we want. What we really want, probably, is to evenly space them along the phylogenetic tree, or possibly weigh ones that are further away from us in time, but are still serve the same function. So those ones are weighted more highly in the scoring. And we've seen with one improvement, an improvement in the correlation in the more recent version of the program. But this is not something that's been done, something I'm interested in. This could also form a paper in and of itself. So kind of the principles of it that I would like to implement is modular design, so that everything is able to be tested independently. It can be tested at any point. So for example, there is a protein design framework available called Rosetta, but everything's tied together, which makes it somewhat difficult to reuse. It also kind of limits some things that can be implemented in it, because it forces everything to use the same role. So it's a program that does docking, it does design, it does protein structure, so it does many things, but to some degree it doesn't do all of them particularly well. So I'd like to have one program with a library attached to it that anybody could use, free to use, that anybody that can be quickly understood. You wouldn't need to understand the entire program to add things to it, and you wouldn't need to understand everything to test specific features. And that goes into the second thing, which is both the design, the principle of design, and the principle of actually designing the program, which is a separation of roles. So in kind of conceiving this project, I'd like to have everything, no person should be stopped waiting for the work of somebody else as much as possible. So I'd like to kind of separate out the roles, which we'll show on the next slide, of what people can do, and so that they can jump in and out, as I expect people who are interested or want to do, get started quickly, and if they get busy with life or something else, they can leave without kind of screwing everybody else up. And the third thing is that it's not overly reliant on machine learning, and it has features that make sense. So the one that I showed with the machine learning, that was more random forest, but that can be cast into kind of a linear model that recapitulates that random forest model, so that we can understand kind of what we're looking at. So a lot of the newer techniques use deep learning and other machine learning techniques where it's very difficult to see how it actually operates. And I think that is not ideal for a lot of cases. And this was designed in kind of a simple way, deliberately, so that it is easy to understand what is going on and therefore easy to modify and kind of easy to interpret. And I'd like to keep that. So if people want to get involved, the easiest thing to do is to join the BioSci research server, which I list here. Introduce yourself in Optimus Spine channel. People are looking for one thing is patience and initiative. It's right now mostly me only working on it. It'll probably change, but the more people that are involved, and it becomes somewhat easier, provided that we have a good structure on it. I don't think that you need a lot of background knowledge to get involved. Some structural biology knowledge is helpful, and some of the things I'm doing, if there are people that are good at math, like at particularly topology and graph theory, there's some things that I'd like to try that would really help us out a lot, and other projects as well if they're interested in collaborating. So the specific roles, as I said, it was kind of dividing it out. So they're overlapping. One person can belong to multiple, how I can see that one person can be in multiple roles simultaneously. So there's some people called Team A working on the individual features. This is the bulk of the program. And from that, we also have a separate team, which can be the same people, but a separate role, working on building the model, the machine learning from it. And so these two are separate, designed to be so that the modeler can work without all the features being designed in the feature, and then the people working on the features can work without the complete model being built. Another thing is designing the server and getting it running. So I'd like to have it both downloadable as a library and available as a web server. The fourth one is integrating with a program called Osprey or OpenMM. So I mentioned we have Foldx. There's more advanced programs available, which I think Osprey is the most advanced one right now used for protein design. And I'd really like to move into that and being able to design our own force field and not rely on Foldx, which is closed source and honestly not really documented well what it's doing. Another option is adding new data into this. Is all based currently, all the programs that are trying this are based on something called Skimpy, which is a database of experimental binding experiments for which there is a crystal structure available as a complex. However, there's also binding experiments where there's no structure available. And there's been new experiments, such as the COVID one, since that database has been released. So the more data we have, the better it does. So some people just looking in the literature for new experiments that we can add to the database would be great. Optimizing the code for the speed critical parts is another great task for people who are interested in programming and not so much necessarily maybe in the structural biology. And also kind of documentation and writing is always great for people who are interested in actually writing the paper. This is kind of how I kind of worked on that before. It's a website called Trello, where we have a board where people get assigned to, and there's a task, and each task then kind of moves from working on to completed and then done. And I can show you kind of example of specific tasks, what they look like. So you can see here, each task will have a description. why it's important and when it's supposed to be done. Attachments, for example, for literature references or other detailed explanations. And then were the more kind of complicated programs, the step-by-step instructions in words on what you need to do to finish this particular feature. And then kind of an activity log of who is doing what and what is done. So that's kind of my vision of what I think that would be great. There's other kind of collaborative projects that if you've kind of been listening in on the bioinformatics channel, I've kind of discussed, for example, benchmarking what AlphaFold can actually do in terms of practical output. Is, for example, how well it does in docking, whether predicted structures from AlphaFold 2 are realistic to use in screening experiments for drugs and other things. There's a few other ones that I mentioned there. And since I think we might be running short on time and imagine people have questions, I'm going to stop here and open it up to the floor. Okay, so can you recommend some resources for things like topology and some of the basics required to understand the project for biologists to get up to speed? The topology was just an example of some of the things that I've been thinking about. I have a list that I've kept on BioCord, I guess it's called Jeff's Big List on the Discord server. I'm going to open up an Optimus bind section on there and start listing things for that in that section fairly soon. So the timeline for this month is actually very busy for me. I'm going to be kind of organizing things this month and then working on the getting it ready in September and also fixing the old server. And hope to start sometime in early September. As far as what kind of time commitment, it's hard to say. Basically, it's whatever you put into it. Whatever you put into the project is kind of what you get out. If you can only do a little bit of time, but you're kind of familiar with the background, you can get involved pretty quickly. If I have to walk you through it, it might be more difficult. And also, as mentioned, one thing that kind of the key thing that we're looking for is leadership. The more people that have kind of serve as leading the individual teams or kind of helping out with the leadership of the project, the much easier it gets. And this also relates to the upper limit of people that you can handle working on Optimus Bind. So those two things are tied together. So if I have a bunch of experienced people, it's not that hard. If I have a bunch of people that need to get everything up to speed, it might be more difficult. And then that goes back into the patient, so that also ties into things go slower and I can't get back to you right away. Is there any other questions? Or any other questions about collaborative projects, how that might work in general?
Hi, Jeff. Yes, we have two questions here from the Discord and YouTube audience. The first one is, can you recommend resources for things like topology and some other basic required to understand the process of the biologist to get up to speed? And
Yeah, that I'll put
that the
on
big
Jeff's
list,
list. Yeah, I'll
right?
put that up soon.
Perfect. And then another Discord user asked, is there an upper limit of people that could get a handle on working on Optimus Bind? I can imagine it could get quite stressful after a point.
Yeah, so as I said, that depends on the experience and how many people are willing to take leadership roles. So
Wonderful.
I can't give a number on that. As far as the paper, authorship gets unwieldy after about 16 people, because then you have to abbreviate it. But pretty much everybody who does something can probably serve as an author up to a point.
Alrighty. So I think those are all the questions that we collected from the audience. We do have a few more minutes left, so if anybody wants to type in any questions quickly. Oh, I just saw one pop up on the summer conference questions. Do you believe that Discord could be used for collaborative projects that are experimental?
Yeah, as always, like the anonymous thing is kind of one of the big obstacles, and there's been discussions on that. It's difficult to collaborate between anonymous people, at least if they don't know each other. But I don't see why you can't just say, I am working on this. I'm looking for a person doing X. I know X. I'm looking for experiment in Y. And I guess DM me or email me if people are comfortable with that. So as I mentioned, there's other projects. Some of them are, if like I said, with the math background, if there's somebody pretty good in math, there's immediate collaborations that would not take that long. That would get you credit on a paper that I can think of right now for stuff that I'm writing.
Right. Yeah. And this is something that we have talked about on the server before, that it's kind of difficult in terms of like credibility, and for that reason you have to forego the anonymity which Discord provides. But I also think that Discord is a wonderful and very malleable platform that allows us to do many more things. We have another question from the audience saying, do you believe that Optimus Bind succeeding could propel Discord as a platform for collaborations? I'm guessing that the question is kind of based on like the success of Slack, probably.
Yeah, so first another comment about anonymity. So like one thing is separating them out into two servers. So the bioscience research one's a very low volume server, so there's not much activity on it. So that kind of makes it easier to focus to some degree. The other thing is if you use a pseudonym there and a pseudonym here, and then your real name on a paper, there's not really a way, unless somebody's really determined, to figure out who you are if you're concerned about that. So we can't guarantee anonymity if you're going to publish under your real name, but having kind of like an extra layer of protection if you're concerned about that helps, like having a second server. As far as everything succeeded, so like one thing that I was interested in was just, can you do this with Discord? So there's kind of two different ways that these types of projects have been done. So one obviously is that things have been done in an academic lab as part of a team. So there's been some disadvantages of that, and if you're not in a lab, you can't do anything. And so when COVID hit and people couldn't get into school, and even computational labs were shut down for a while, it kind of set a lot of people kind of adrift, and we've seen a lot of that in kind of the talk of the server. I want to start school, I want to start studying, I want to do things, but I can't because nothing's available. So this is kind of giving a way for people who've been shut out to participate. And I think that if we can demonstrate this project as a success, so if we have it affiliated officially with BioCord, like as if it does get published, and I think it will. I mean, it certain things like the actual servers kind of it will almost certainly get published, probably within the year. I'm not sure, but at some point it definitely will. It gives kind of an example, and we just kind of have to publicize that, like, hey, you can actually use a kind of a collaborative community to do this, like, a project coming from the community rather than an open source one that people attach themselves to, or a lab that people work at and then join. Yeah, I mean, you don't need an affiliate. Question about whether you need an affiliation. You don't need any affiliation at all. It's specifically to help people without affiliations work in this kind of work. Is there any other questions regarding the BioCord, its relationship to these projects, the resources available, some of the questions not a biologist asked on the questions channel? All right, then I gave out the link. It's in the resources, I think, the Biopsy Research server, and just checking that channel, let me know you're there. And then, like I said, this month is pretty busy. I will be working on it more intently at the beginning of starting September, and I'll be organizing in the meantime.
Alrighty, thank you so much, Dr. Brender. That was a wonderful talk. I think that from the audience, everybody was really excited and commenting on how such a great opportunity it would be and how big brain it sounds. But yeah, thank you for sharing with us, and we will see you around the server. And if anybody has any further questions, you can always just tag Dr. Brender on the Discord, and
it's
he
easier.
likes to answer.
I'm not a good speaker. I'm much better at writing.
Go ahead.
So just tag me either there or on the other server. If it's in-depth question, maybe the other server might be better, but the regular BioCord server is good too.
Alrighty, thank you so much, Dr. Brender.
Yeah, thank you.